Welcome. Okay, so you've sent us down a really fascinating path this time. We've been digging into these sources on Genesis machines.
That's
The
right.
whole idea of actually creating life basically from scratch.
Yeah,
Well, it's pretty mind-bending stuff.
it really is. And the materials you sent highlight just how ambitious this field has become. We're talking about potentially engineering life itself.
Right. So let's get straight to it then. Based on what we've read, what exactly are these Genesis machines? What's the core concept?
Well, fundamentally, the term refers to systems, technologies, designed to generate living organisms from non-living matter.
Okay, so like abiogenesis, how life started originally, but engineered.
Precisely. It's taking that ancient question, but moving it into the lab. Scientists are trying to, understand and actually control the steps involved.
So trying to speed it up, maybe even direct how life could be made?
Exactly. It's like trying to, well, direct the movie of life's origins, not just watch it.
Gotcha. And one of the sources pointed to a really key moment back in 2010.
Ah, yes, the J. Craig Venter Institute. That was definitely a landmark. They chemically synthesized the entire genetic code, the whole genome of a bacterium.
Mycoplasma mycoids, I think it was.
That's the one. And the really incredible part was taking that completely artificial DNA
Yeah.
and inserting it into a host bacterial cell. Basically, they gave the cell a whole new operating system that was designed on a computer.
And that result was Cynthia. The source called it the first organism with a fully human-made genetic code, like a biological software upload.
That's a great way to put it. It showed we could not just read the code of life, but write it and, crucially, make it run.
Wow. Okay. Now, the sources also talked about two main ways scientists are going about this.
Yes, the top-down and the bottom-up approaches, they're quite different strategies.
Right. Can you break those down for us? What's the difference?
Sure, so the top-down method involves starting with existing life and simplifying it.
Taking something alive and stripping it back.
Exactly. The Mycoplasma Minimal Genome Project is a prime example. They systematically remove genes to figure out the absolute minimum set needed for life.
Like finding the core operating system of a cell.
Precisely. What are the bare essential instructions?
Mhm.
It's about understanding life by dissection almost.
Okay, that makes sense. Taking it apart to see what's critical.
And then you have the bottom-up approach. This is really where the idea of creating life entirely from scratch, these Genesis machines, comes into its own.
Right. This involves building protocells, is that the term?
Yes, cotocells.
Mm.
Think of them as really primitive pre-cellular structures.
So not quite cells yet, but trying to get there.
Sort of. Researchers use things like lipid membranes, fatty molecules to form tiny compartments like a basic cell boundary.
Okay.
And inside those, they put molecules like RNA and peptides, key building blocks for genetics and function. The goal, as some of the research like Adamol and Sostak showed, is to get these structures to carry out basic life processes.
Like metabolism or even replication.
Exactly. Things like that. But crucially, doing it without starting from an existing biological cell, assembling it from components.
So not simplifying existing life, but building something lifelike from basic chemistry. One source called it Life 2.0, engineered, not evolved.
That really captures the distinction, doesn't it?
Mhm.
It's a profound shift in thinking.
It seems like it. And this opens up, well, a huge range of possibilities, according to the sources.
Oh, absolutely. The motivations here go way beyond just curiosity about life's origins, so that's certainly part of it.
Yeah, the practical applications mentioned seem pretty significant. Things like designing organisms to make specific drugs.
Yes, or to clean up pollution, maybe even capture atmospheric carbon.
Using engineered life to tackle major global challenges.
That's a huge driver.
Yeah.
And then there's the idea of engineering life for extreme environments, like Mars, for instance.
Which has massive implications for space exploration and finding life elsewhere.
Definitely. And also exploring different kinds of life chemistry. Could life exist without DNA or RNA? Some researchers, like Benner, are looking into that,
Yeah.
using different molecules altogether.
So expanding our whole concept of what life could be, chemically speaking, there was also a more recent development mentioned from 2021.
Right, the Pelletier group's work. They managed to create a synthetic bacterium that could actually grow and divide.
So not just making the parts, but building something that could sustain and, well, reproduce itself.
It was another big step. Shows we're getting closer to constructing truly autonomous artificial life.
The potential seems incredible, but the sources also spend a lot of time on the flip side, the concerns.
And rightly so. With this kind of power comes some very serious questions and responsibilities.
Like, fundamentally, is synthetic life even real life?
That's a big philosophical one. How do we define it? How should we value it if we create it?
And then the more practical safety concerns.
Absolutely. The potential for misuse is obviously there. Could someone engineer harmful pathogens? That's a major biosecurity worry.
Yeah.
And even unintended consequences. How might engineered organisms interact with natural ecosystems if they ever got out? We need really robust safety measures and containment.
It sounds like there's a real need for careful oversight and ethical guidelines, though the sources also mention worries about regulation keeping up with the science.
That's always the challenge with cutting-edge tech, isn't it? There's also the question of, well, rights for synthetic organisms, potentially. And could this technology widen societal gaps? Lots to unpack.
These are definitely complex, heavy issues that need ongoing debate as the science moves forward.
They really are. It forces us to confront basic ideas about biology, creation, and our place in the world.
So, putting it all together, understanding life's origins, engineering solutions, but also these deep ethical and safety considerations, it really makes you stop and think. If we gain the ability to create life, how does that change our view of what it means to be alive? Does it change how we value the life that evolved naturally around us? That's something to chew on.
It certainly is. A conversation that's really just beginning, I think, and one that will evolve right alongside the science.
Well, thank you for walking us through all this. This deep dive into Genesis machines has definitely given us, and hopefully our listeners, a lot to think about.
My pleasure. It's a fascinating and incredibly important area.